Metabolism deals with the interactions of the biomolecules in a cell and chemicals from the external environment. By manipulation of these interactions, a cell can be redirected to biosynthesize chemicals of interest or break them down. Metabolism is typically separated into two topics, catabolism and anabolism. In catabolism, chemicals from the environment such as glucose are deconstructed enzymatically into a set of metabolites that are the primary metabolites of the cell. This set of chemicals is largely conserved across all organisms and defines the chemical state of the cell. In anabolism, new molecules are constructed from the primary metabolites. These include natural products, polymers, and biotechnology targets such as biofuels. Enzymes define a class of molecules that exhibit cyclical state diagrams, catalyze reactions, and are associated with the domain of living systems. Usually they are proteins or complexes of several proteins. However, there are many examples of RNA-only and RNA-protein complexes, particularly for reactions that involve nucleic acid substrates. There are several general characteristics of enzymes you should understand. In general, enzymatic reactions are very specific to their substrate in terms of what molecules they will bind to, where on those molecules they will perform chemistry, and the side of the molecule they will interact with. This gives rise to strict stereoselectivity, which is the preference for one stereoisomer over another, and regioselectivity, which is the preference for one functional group on a molecule over another functional group. Though there is not yet a comprehensive survey of enzyme specificity to make such broad sweeping claims, the general experience is that anabolic enzymes tend to be more specific for their substrates than the catabolic enzymes. Often catabolic enzymes are involved in breaking down insoluble or polymeric substrates, and they have a broad substrate specificity to deal with the many use cases they encounter in the environment. The enzymes involved in interconversions between primary metabolites are a mixed bag. Some are very precise and others very promiscuous. For example, during amino acid biosynthesis, the last step is this transamination reaction. Though there are only 3 enzymes capable of doing this chemistry in the cell, that set of enzymes can handle all 20 natural and most unnatural alpha-keto acids. If we go back to our biochemical diagram of a bi-bi enzymatic reaction, we can capture this promiscuity primarily in terms of loose constraints on the non-covalent reactions. In general, the chemistry of the covalent reaction of an enzyme is specific, but there will be degeneracy in the compounds that can bind to the enzyme and participate in that reaction mechanism. Largely, it reflects a diversity in shape preference for the enzyme, but the precise molecular transition taking place is quite exact. Traditionally, enzymes have been exploited for biotechnology purposes in all sorts of ways. The simplest of that is biocatalysis, in which the purified enzyme is reacted with a substrate. Such techniques have been thoroughly industrialized and approached from a chemical engineering perspective. There are many issues with the cost of the enzymes, the methods for recycling the catalyst, and methods for dealing with the fact that many of the substrates are hydrophobic, but the enzyme only likes to work in an aqueous environment. These issues are often addressed by compartmentalization strategies such as immobilizing the enzyme in micelles or liposomes, separation of the enzyme from the substrate chamber by membranes, or by forming large aggregates of the enzyme using polymerization chemistries. A biotransformation is much like a biocatalysis in the sense that it is a chemical process that treats the enzyme-containing sample as a traditional catalyst to convert one substrate to another. In a biotransformation, whole cells are used as the catalyst, and due to their complexity, a large number of reactions are often observed. The cells are not growing under the conditions of the experiment and can be thought of as a limiting case of compartmentalization strategies such as those used for biocatalysis. Often it is necessary to further compartmentalize cells as alginate beads or other means for an efficient process, but many reactions such as oxidations and lipase-style transformations can be easily performed by specific combinations of substrate and organism. Here we show stereoselective and regioselective reduction of ketones in a substrate by biotransformation with Baker's yeast. Traditionally, enzymes are classified according to EC numbers. These 6 general categories of reactions loosely correlates with reaction mechanisms. Oxidoreductases catalyze the oxidation and reduction reactions. As a class, they include NADH, FADH2, oxygen, and various other cofactors. The class is defined by the change in oxidation state of atoms during the transformation. Here, an alcohol is being oxidized to a ketone. The electrons are transferred to NAD+ molecules, which is reduced to NADH. Transferases transfer entire moieties from one molecule to another. You can think of the term moiety as being one level up from a functional group. They correspond to small unique carbon skeletons that are frequently observed in biomolecules as intact subgraphs. Transferases typically transfer a methyl group, a phosphate, a sugar, or an acyl group from one chemical to another. Hydrolases are, crudely, the reverse reaction of a transferase. They cleave heteroatom carbon bonds and thus separate carbon subgraphs within a larger molecule by reaction specifically with water. Lyases cleave bonds by means other than reaction with water. They often involve the breaking of carbon-carbon bonds. Here we show the elimination of carbon dioxide, which breaks a carbon-carbon bond. Note that no redox happened here, but a carbon skeleton was altered due to the reaction. Isomerases alter a molecule in a way that does not change its molecular weight. There are epimerases, such as the one shown here, that flips a stereocenter on the substrate. In other situations, an atom might be moved from one carbon to another. Mechanistically, this is the most poorly defined EC class since many different mechanisms can result in this general characteristic of the substrate and product being isomers of one another. In this example, an NAD+ dependent redox reaction is taking place, but since the net change in oxidation state of the substrate is 0, it is classified as an isomerase instead of an oxidoreductase. Ligases are very similar to transferases in the sense that they involve the transfer of carbon skeleton moieties from one set of molecules to another set. They are typified by the coupling of the reaction mechanism to the hydrolysis of ATP, which drives an otherwise thermodynamically unfavorable reaction towards the products.